So it is my great pleasure to introduce our first speaker for the semester, Professor Shelle Geilholz. Probably many of you know Shelle. She is a professor at BME in Emory at George Tech. She did her bachelor's in the University of Missouri Rolla, and then her PhD in University of Virginia. They were both in physics. So she studied MRI, and then she went to do a postdoc at NIH where she got into functional neuroimaging, so fMRI, and now she is working on fMRI in both humans and rats, which is a really amazing combination where you get to study these systems in a way that very few labs around the world can. So really looking forward to your talk, Shelle. Take it away.
Oh, thank you for that great introduction, Dobie. Let me just share my screen here.
Oh,
and thank you both, Dobie and Ming, for inviting me today. I'm really excited to have the chance to come and talk to you all. Thank you all for coming. I hope we'll have time for a great discussion at the end of the talk, and so I'm going to try really hard not to talk too long. So I want to start out by giving a little bit of the big picture for the work that we do and talk about the problems that we're trying to solve, what we're trying to learn, and the tools that we use to do it before I really dive into this particular project. So I got interested in the brain. That's a long story, but it happened. And the thing I think is really cool about the brain is that activity occurs at so many different scales. You have, transmission at single synapses. You have these localized kind of mesoscale structures like columns and layers that are more specialized. Then you have areas that do specific tasks like process sensory information. On top of this, you've got these sort of widespread inputs from neuromodulatory systems that deal with things like arousal and attention. And so all of this is working together. And it ranges, from spatial scales at a few microns all the way up to centimeters, and at temporal scales from milliseconds for action potentials to changes that occur over hours or days or even over a lifetime. So when you put all this together, the brain really looks like kind of this, epitome of a complex system. And physicists find complex systems really interesting. And so that's kind of what got me excited about it. So when I talk about the brain, I like to compare it to another complex system that we're all familiar with. And that's, the weather, or if you think long-term, the climate. So you can think about, measuring the temperature and the precipitation and the humidity and the barometric pressure all at one particular place, say my office at Emory in Atlanta, Georgia, at a particular day and a particular time. And a lot of times that's what we do when we're studying the brain. We look at an area, we look at spike rates and how particular neurons respond to a particular stimulus. But if you really want to understand and predict what the weather is going to be like at a place in the future, you kind of have to look at the weather as a whole and make these sort of complex systems models that let you say, oh, in 3 days from now, it's likely to rain. And so that's what we want to be able to do with the brain, to kind of be able to define the weather or the climate of the brain and how that influences what's going on at the more local level. So, we might talk about high pressure areas or low pressure areas for weather. I don't think we're even clear on what that would mean for the brain yet, but we need to find ways that describe this. And especially if we want to talk about some sort of dysfunction that might be related to, say, a psychiatric disorder or a neurodegenerative disorder, maybe that's something more like a storm, some big event that we need to be able to predict. So the downside, to the comparison to the weather is there's not a lot we can do about the weather, right? If it's supposed to rain in 3 days, well, we don't really have a way to head it off. But with the brain, we've got tools that are available and more tools that are coming that give us this possibility of manipulating brain activity at different areas, on different time scales. And so I think understanding the systems level, the activity patterns of the brain at this level will help us to be able to predict how people respond to a particular stimulus. So it's really interesting. Even if you take a very stable system like an anesthetized animal, let's say we've got a deeply anesthetized rat, and we give an electrical stimulation to the forepaw 3 times in a row. Every time we give that stimulation, we'll get a different neural response. So there's this inherent amount of noise and variability. Some of that is present, at the level of this processing the incoming stimulation, but we think some of this is also present because you have this systems level impact on what's going on at that individual location. And so we think trying to understand the brain at a systems level will help us to predict how the brain responds differently to the same stimuli at different times. And if you extrapolate that a little bit further, you can say, well, maybe then we can also explain individual variability in behavior and cognition. We want to know why certain people are better at certain tasks, but we also want to know why the same person is better at the same task at different times than at other times. And these are all things we're still trying to figure out with the brain, where I think a systems level approach can really help us move that understanding forward. And then kind of the holy grail, of this approach to looking at the brain would be to define normal patterns of activity at the systems level, things that characterize a healthy brain, and find ways then to use these interventional methods we have, like transcranial magnetic stimulation, TDCS, all of these different tools that we have to drive disordered activity back to those healthy patterns. But we really think that this is going to have to be done looking at the brain as a whole rather than looking at activity in one individual place. Okay, so that's what we want to do. How do we get the data? How do we look at activity throughout the brain at a systems level? And if we're talking about healthy human volunteers, we have a limited number of options. So first of all, if we think about neural activity, we usually think about things that are related to the neurons themselves, the firing rate, transmission at synapses, so electrical activity, chemical activity, and those are the kind of things that we can pick up with EEG or MEG from outside the brain. Now, those have great temporal resolution. They're directly sensitive to this type of neural activity. The spatial resolution is not so good. We're talking more like a centimeter or so, and you lose sensitivity as you get away from the cortex. So it's very hard to study deep brain areas that can be important for this whole brain organization of activity using these methods. So if we go further down the chain, if you have more neural activity, you also need to have an increase in metabolism to support that. So you use more glucose, you use more ATP, you need more oxygen, and your brain doesn't store energy. So when you need these substrates, we have an increase in blood flow to take them to the places where they're needed. Blood flow, blood volume, and also blood oxygenation. The metabolic changes are slower than the electrical or chemical changes, and the hemodynamic changes are slower still. They occur on the order of seconds. But if we are willing to step away a little bit from the neural activity, we can look at these metabolic changes especially very well with PET, positron emission tomography. Now that does require that we inject a radionuclide, but it has decent spatial resolution, on the order of a few millimeters. And the temporal resolution, on the other hand, is very slow, usually minutes, maybe even tens of minutes. That's kind of the downside of PET. So then we come to my favorite, fMRI. And fMRI is mostly sensitive to the hemodynamic aspect of this neural activity. Now, I like fMRI because it's completely non-invasive. We don't have to put in that radio tracer like we do for PET. The spatial resolution, depending on how you set up your scan, can be on the order of a millimeter. Maybe better if you focus in on a small area, but say a millimeter or so for whole brain imaging. And the temporal resolution is on the order of seconds. Again, we've got a little wiggle room. We can go faster if we need to for a smaller area. But looking at seconds and at millimeter level activity gets us to the point where we're sensitive to those areas of the brain that specialize in different aspects of brain function, and we can look at the response in those areas on a time that's kind of on a scale of, the type of tasks and decisions we make in the course of a day. So just really quickly to talk a minute about how we actually get contrast from this with MRI. MRI is sensitive to changes in the local magnetic field. And deoxyhemoglobin is paramagnetic, which means it really disrupts the magnetic field. Oxyhemoglobin is dimagnetic. When we have activation, we are going to have a change in the ratio of the deoxyhemoglobin to the oxyhemoglobin. Now, you would think that you're using more energy and so that we'd have more deoxyhemoglobin, but in fact, the brain is so good at supplying those active areas with additional blood flow that we get an increase in oxyhemoglobin instead. And so this is showing a typical blood vessel. These blue dots are our deoxygenated hemoglobins, and these lines are showing you how far out this magnetic field perturbation might go. That's before activation. So when we have activation then, when we have that increase of blood flow to supply that area, we have less deoxyhemoglobin, more oxyhemoglobin, and we have less disruption to that magnetic field. And what that means is that our MRI signal goes up. And so it looks something like this. If we're taking an image at, say, every second, and at some time during the scan, we turn on, say, our 4-pass stimulation for a rat, we can look at different areas over time and see how the signal changes during that task or stimulation. So my animation doesn't seem to have worked here, but if you follow the green line, it's just kind of messy. It doesn't show any particular change during the time of that stimulus, indicated by this red bar. But if you follow the blue line, you can see that whenever the stimulus comes on, we get a little increase in signal, and when it goes off, we get a decrease. And so there's a change that's linked to the application of that stimulus. And because we know the timing of that stimulus, we can determine which areas show that change. And we do this maybe by correlation with a time course that tells us when the stimulus is on or off. We can do tricks like average during the stimulus and average when there's no stimulus and do a subtraction. Lots of ways to figure out which areas change during that stimulation. Now, when you give a stimulus or a task, you have a very small change in activity, and it accounts for a very minor amount of the overall activity of the brain that's already intrinsically happening. And so my group is really interested in studying this intrinsic activity. So not what happens during a particular stimulus, but what is going on in the background that that stimulus gets added onto. And so we do something called resting state fMRI. And all that means is that we do the same thing as regular fMRI, but now there's no task. And this is kind of handy sometimes because people don't have to cooperate. For example, if you have patients who are having trouble doing a task or something, if you just do this resting state scan, they don't have to actually do anything. We're just putting them in the scanner and tell them to relax and look at a dot in the center of the screen. So the idea is that using this technique, we can map this intrinsic activity across the whole brain, non-invasively, which is awesome. Now, the problem is that we don't know the timing of a task or stimulation, so we don't really know, there's not a particular time to look at to see how things change. We've got thousands of voxels, about 1,000 time points, and somehow we need to summarize all of this information into something that we can interpret and compare across groups. So just to go back to the brain for a second, we talked about all of these different scales, these different temporal scales and spatial scales. Well, I want us to remember that all of that is getting mashed together now into this resting state fMRI signal. So we can't distinguish, synapse level activity from area level activity from neuromodulatory activity. It's all just stuffed into the signal that we're pulling out with the MRI that's looking at fluctuations in oxygenation. And like I said, we don't have any idea about the timing, so we can't do our trick before where we look at changes before and after a stimulus is applied. So we've got to come up with something else. And the way that people have been analyzing this data for about 20 years now is to look at correlation. So the idea is that we've got this chain of events in two different areas, right? So we have neural activity, which leads to metabolism, which leads to these changes in hemodynamics, which leads to our MRI signal, also known as the bold signal for blood oxygenation level dependence. So we've got that from one area, we've got that from the other area, And so if the MRI signals from these two areas are coordinated or correlated, we usually use Pearson's correlation to measure this. There are other techniques, but they give similar results. If those signals are correlated, it implies that the neural activity in these areas might also be correlated, and that these areas are communicating with each other to somehow coordinate their activity. And so people refer to that as functional connectivity. Okay, so I said we've got this resting state fMRI data that we acquire over time without a stimulus. I said we can look at correlation, and here's one way that we can do that. Maybe we put a seed in a certain area, like where you see this high correlation, and we look for which other areas correlate with that. And so you can see we can get these different areas across the brain that all seem to be communicating that we would call a functional network or functional connections. So there we're looking at correlation that persists over the entire scan, which is usually 10 minutes or so in humans. But you can imagine that we might also want to see dynamics. I said we want to study the brain as a complex system, right? So if we're getting these static measurements of the brain, that's really not telling us much. That's kind of boring, and it doesn't let us get at the dynamics that really distinguish the different scales of these systems. And so we're looking at a lot of these different dynamic approaches as well that build on this average or static approach to analyzing the data. And I'm going to come back to those in just a minute. The last thing I wanted to show you about the average functional connectivity is a way that we often use to look at it. So to reduce the data dimensions, I said that we can have, tens of thousands of voxels. A lot of times we register the brain to an atlas, and then we divide those voxels up into areas that have kind of similar functions. So like here you can see sensory and motor cortex have been put in their own groups. The visual areas are divided into a few different groups. And so that lets us parcellate the brain. We kind of average the signal from those areas. And then if we want to, instead of choosing a single seed, we can calculate the correlation between every area and every other area and make a matrix like this one shown over here. Now this has been sorted so that areas that are kind of within the same general functional network are grouped together. And so you see these functional networks as these areas of high correlation along the diagonal. So you'll see more of these correlation matrices as we go on. But as I said, that was average functional connectivity. So the data I just showed you was from the Human Connectome project, which has something like 1,000 subjects. For each subject, we have about 6,000 time points and 100,000 voxels. And we've taken all of this data in that prior slide, and we've condensed it down to a 360 by 360 matrix. We've just thrown away all of the interesting stuff. All right, maybe not all of it, but we've thrown away a lot of information. And I said that we want to see how this changes over time. Why? Because we know your brain's not doing the same thing over time. If we put you in the scanner, you're going to be laying there thinking, gosh, I'm bored. When are we getting out? I want to go have lunch. This is getting uncomfortable. Or maybe you're getting sleepy. That's what happens to me. I always fall asleep in the scanner. And so all of these changes are kind of layered over that resting state fMRI data. We also know from other studies that changes in functional connectivity can relate to behavior. So not only are these interesting just in terms of how the brain is organized, but they actually have real world applications to how we behave. And so we really need to capture the dynamics, and that brings me back to our approaches for time varying analysis. So I'm not going to go into these in detail today. My lab has done a lot of work on different methods for looking at time varying functional connectivity. But just to give you a kind of an overview, the simplest thing you can imagine is to take that 10-minute long scan and chop it up into chunks, right? So maybe instead of looking at correlation for the whole 10 minutes, we look at it for 1 minute at a time. And so we get a time course of correlation. And that's called sliding window correlation. And there are a lot of ways to do that and use different windowing techniques. Another thing that you might think about is looking for some sort of repeated spatiotemporal pattern. So to go back to weather for a minute, we know there are these La Nina and El Nino periods that have kind of their own characteristics and that they come up every so many years. They show up again and again over time. maybe there's something similar for the brain. And there is, and I'll tell you more about that one in a second. And then kind of the last of these approaches is to look at coactivation patterns. And so these are mapped time point by time point during the scan. So you might say, okay, I'm going to look at the times when somatosensory cortex is, within its top 10% of activation, and I'm going to see what else activates with that, what co-activates. And so you can make these patterns of areas that tend to co-activate together and try to discern how these networks are interacting with each other over time. Okay, so I said that I was going to talk more about quasi-periodic patterns because that's what I'm going to use as my example for today. We've done a lot of work with this, and I mentioned this kind of like El Nino or La Nina. We didn't go looking for weather cycles in the brain data. This was something we stumbled across. One of my first students and I were talking, and I said, we really need to figure out how to look at dynamics. And he made a movie of brain activity. And we watched it and we said, whoa, there's this cool pattern that keeps happening. This was for rat data. And we would see activity kind of start at the lateral sides of the brain and move towards the medial parts. We said, that's really strange. So he made a pattern finding algorithm that would go in and pull out that pattern. And it turned out that that pattern is so pervasive that we don't even have to identify an initial starting point. You can just kind of randomly select one and you'll come out with the same pattern. And you can do the same thing in human data. And we get a really interesting pattern there that I'm going to show you in just a minute. But what our algorithm gets us is this pattern template. Now this is a 4D template. We have volume, and for each volume it tells us how active a particular area is at that time point. And then we have time, so 3D by time. And these patterns last about 20 seconds in people. And then we also have a time course that is the length of our original scan that tells us the correlation between this template and the original scan at any point in time. And so when you have an occurrence of this pattern, you get this nice strong peak that tells us when that pattern occurs really strongly. Okay, so what do these look like? I'm going to play this movie for you in just a second, and this is the template that we got from that Human Connectome project data. So first, just a little of interpretation. Warm colors mean the activity is above average at that point in the pattern. Cool colors mean it's below average at that point in the pattern. And what you see at this very beginning is interesting in and of itself because it contains two networks that have been described ever since people started looking at resting state fMRI. One is the default mode network, which is currently in blue, and that has been associated with some sort of internal focus, mind wandering, not doing anything in particular. The other network, sometimes called the task positive network, is in the warm colors, and that's been associated with external focus, or being active during a lot of different types of tasks. And so you can see these are kind of opposite of each other, and it's known that these networks are anti-correlated. And so when I play this movie, you'll see that in this spatiotemporal pattern, they're also anti-correlated, which makes perfect sense. So here we go, and so you see that they come up very strongly, first active and then deactivated. So we started with the default mode network deactivated, now it's activated, and the task positive network is deactivated at the end. So I'll play that one more time. Again, this is sped up. This takes about 20 seconds in real time. And you can kind of see these subcortical areas over here, but to convince you that this really is a whole brain pattern, we've zoomed in a little bit on these subcortical areas so you can see the pattern better. And we find, as before, that there's this propagation from one area to another, and that areas that we know are correlated with the default mode network are correlated also during this pattern. So this fits with everything we knew about these networks before. So we found more evidence that these patterns are really interesting, and that comes from a study that defined something they called functional connectivity gradients in the brain. And basically what they did is they looked at the functional connectivity of all these different areas and then kind of arrange them by similarity using dimensionality reduction, so that areas that are highly similar are at one end of the axis in red, and areas that are completely dissimilar are at the other end of the axis in blue. And you might realize that this looks just exactly like that first phase of our QPP, which I've shown over here for comparison. And so it turns out that our patterns sweep across these gradients. Now I said QPP, I don't know if I explained that. We call these patterns quasi-periodic patterns because they show up so often, but we didn't want to say they were exactly periodic because we seem to lose them some of the time. We don't really know why yet. But if I say QPP, I'm referring to that pattern template. Now, this is really interesting because the primary axis also goes from sensory and motor areas to association areas. So kind of, they're opposite ends of the processing hierarchy. They're at the greatest topographic distances in the brain, and they're also at opposite ends of the developmental axis as the brain develops, in utero. So it seems like there's kind of something fundamental about this pattern and maybe about these dynamics that we're pulling out. Now, I've only talked about the primary gradient. There are also secondary gradients going all the way up to gradient 5. And we also have secondary QPPs that match up to these gradients as well. But I'm not going to talk more about those today, really. So we wanted to see how QPPs and this average functional connectivity were related, just to understand things better. And so this is that matrix I showed you before, where we have 360 areas by 360 areas, high correlation in red, anti-correlation in blue. Now, this would normally be symmetric because we're calculating correlation, but in the top right matrix here, I'm showing the original correlation, and here is the correlation at bottom left after we've regressed out that primary QPP. And you see we lose a lot of the anti-correlation and a lot of the correlation within the networks. And by the time we take out the first 3 QPPs, there's very little correlation left. It's mostly between left and right areas that are exactly homologous, like left and right primary motor cortex, for example. So that's really interesting because it says these 3 patterns then explain most of this average functional connectivity. And I think that's a great motivation to go on looking at these dynamics. So what we've got is this whole brain pattern of coordinated activity. But what does it mean? Well, we don't know. We were afraid it was hemodynamic because I mentioned that when we look at the bold signal, we're looking at this hemodynamic response. So we set up some multimodal imaging studies. And this first one that we tried to do was simultaneous electrophysiology and MRI. Now, there's a story behind that, and it was quite a challenge. You can see this is one of our early images up here at the top, and you've got this big black hole where we stuck a metal electrode into the brain. You can imagine putting metal in messes up the magnetic field and messes up our image. Just as badly, when we're trying to record the local field potentials, anytime we acquire the image, we get these big spikes that eliminate the local field potentials, and we have to wait for a little while before our amplifiers reset before we can get good data. But we managed to take care of this. So this image down here at the bottom left actually has 2 electrodes implanted, one on either side, but you see the image quality is still pretty good. And after we tweaked our analysis a little bit, we were able to get about 450 milliseconds out of every 500 milliseconds, clean enough to record LFPs. So we decided that was pretty good too. So the first thing we did was just look for coherence between the low frequency electrical activity and the MRI signal. And because we weren't very expert at electrophysiology, we didn't know a lot about what we were doing, we looked at those actual slow measurements. So electrical activity in the 0.1 hertz, which is kind of where these bold fluctuations tend to fall, is right around that frequency. And we found high coherence between the two. I'm showing this for 2 different anesthetics because this work is in rats, and I'm sorry, I do jump back and forth between rats and humans when I give this talk. So sometimes I have a little key at the bottom, which species it's from, but I must have left it off this one. But this is in rats. And so we do 2 different anesthetics to control for the effects that the anesthetics itself has on both the neural activity and on the vasculature. But either way, we get a nice strong band of coherence between the electrical signal and the MRI signal. More interestingly, when we looked at the correlation between the signal from the electrode itself and the MRI signal at different time delays to try to account for that hemodynamic response, we started seeing something that looked like this QPP in the rat. And you can see it most clearly in this data for dexmedetomidine, where you start to see a bit of high correlation coming in the side, gets stronger, moves towards the medial areas, and then we start to see this negative correlation that also moves towards medial areas. And out here again, you can see a little bit of the positive cycle beginning again. So this is saying that that signal from the electrode does correlate with something sort of cyclical that's going on. And now I'm going to show you that time lag correlation between one electrode and the MRI data, followed by the template that we made from the MRI data alone, because I think these are incredibly similar, and I think that's very strong evidence that we're pulling out propagation of this infraslow electrical activity. Okay, so this is the correlation between the simultaneously recorded infraslow LFP signal and the MRI. So it's negative, then it's positive and moving towards the center. Now negative again and moving towards the center. And now positive again. Okay, so now here's the same thing. It's going to be much shorter, but this is just the template we made from the dexmedetomidine MRI data. And you see it looks very similar. And in fact, if we go ahead and do some sort of correlation and check for significance, we get significant correlation not only at the normal hemodynamic delay, but also at time points before and after that again show the sort of cyclicity of this property. But I'm not going to go into that now. So we've convinced ourselves that we're looking at propagation of infraslow electrical activity, but nobody understands infraslow electrical activity very well either, and we don't really know why it would show these sort of coordinated levels of activity and coordinated propagation across areas. So we were trying to understand why this might be, and we came up with 2 ideas. The first is that we know the brain has a structural network, right? You know, any given neuron is connected to another neuron through its axon and the dendrites. So we have kind of this structural network underlying all of this information. And so maybe we've got a sort of resonance or something of that network. Another possibility we came up with after talking to some of our friends who are good at neurophysiology is that maybe there are some of these deep brain nuclei that could modulate the whole brain at once to kind of drive these patterns. And so there are a lot of these systems that use norepinephrine or serotonin or dopamine, and they have widespread projections that might possibly be able to coordinate this. Okay, so to look at the first hypothesis, we decided to make a brain network model. And the way this works is you take the structural network of the brain, which you can measure with something like diffusion-weighted MRI to figure out how many fibers go between areas, or you can use viral tracing. It just depends on your species. But anyway, so we take the structural network, and at each one of these nodes, you put a set of differential equations that describe how the activity at that node behaves, and it's coupled to other nodes through that structural network. So it gets input from the nodes that it's connected with. And so we put this together, we set it up, we let it run, and then we ran our same analysis to see if we could get something that looked like these quasi-periodic patterns out of this model. So here are the results. This was done on human data in this case, and we're looking at things a little differently now, so I need to reorient you. This is the QPP template, but we flattened it out to make it easier to compare rather than looking at it on the whole brain. So each row here is a different brain area. And now high activity is yellow, low activity is blue, and this is the 20 seconds of the QPP. So you can follow, for example, down here in this last area, it gets first high activity, then it starts to switch about midway through, and then it's lower than average activity for the rest of the QPP template. So that's what we're looking at. This is the one that we got from the actual data. And these are the ones we got from 2 different brain network models. And it's interesting because you do see some of that 2-state sort of behavior, but it's much less complex than it is in the real data. And it's more of kind of this flip. You have these anti-correlated networks that just flip. So it gives a hint that maybe there's something to this, this resonance idea, but it doesn't explain everything. So then we tried to investigate the other hypothesis, this idea that maybe there were neuromodulatory inputs that were driving this. And, we were enthusiastic about this because we already knew that pharmacological manipulation of many of these different systems changes the functional connectivity strength and clustering. We know that these patterns change in anesthesia and that these nuclei can modulate arousal levels, so that fit. We know that one of these nuclei that we decided to focus on, the locus coeruleus, in particular is involved in task performance and this trial-to-trial variability. And part of the QPP that I showed you, that default mode network, has also been implicated in task performance and variability, so that kind of fit. There are studies that show that stimulation of the locus coeruleus alters the infraflow activity in the cortex, and we know that they're linked to QPPs there. And then finally, the locus coeruleus is also the site of early alterations in a lot of neurodegenerative diseases. And this is another thing I'm not going to tell you about today, but we found that in mice, using these QPPs lets us classify the Alzheimer's model better than just using static connectivity. So that was another connection that made us excited about this possibility. So the first thing we did is just go back and look at the timing of the QPPs across different areas of the brain. So what I'm showing now is the number of voxels that peak at any given point in the QPP cycle. And so this is a lot of information, but let's just focus in on a couple of key areas. So these 2 lines tell us when the left posterior cingulate cortex, which is a key node of the default mode network, either has its peak deactivation or its peak activation. And what I want you to realize is that if we look at where the peak is, there are areas in the thalamus and the brainstem and deep brain that peak before that cortical area. And so it seems like maybe those are good candidates for driving this activity. And that fits with our idea of, deep brain nuclei like the locus coeruleus being involved in this. So the next thing that we could do that was really simple is to look at, whether these patterns, these QPPs, would change when we had somebody doing a task that made them pay attention, because we said that these neuromodulators are involved in arousal levels. So we looked at the same pattern when people were just resting or when they had to pay attention to a small dot that changed colors every so often and press a button as fast as they could when that happened. And we didn't see any change, so that was a little disappointing. But then we looked at the reaction time and how the reaction time lined up with the phase of the QPP. And we found that if you were in the phase of the QPP where the DMN is active, your reaction time was a whole lot more variable than it was in the phase where the task-positive network was active. So that again said, well, maybe we're still onto something. And we thought maybe it's because the PVT, this task where you watch this dot and press a button, maybe it's not cognitively demanding enough. And so we did a little bit further analysis that I don't have a good figure for yet, looking at HCP data where you had to do a working memory task that made you think constantly about what you'd seen, in the last image. And there we did see that the QPPs are stronger and more frequent than they are at resting state, which would be consistent with an increased level of arousal. Okay, so the last thing that we did in our animal model was to give some rats DSP4, which selectively kills off locus coeruleus neurons. And that was very exciting because when we did this, it decreases the LC input to about 10% of what it normally is. And after that, we couldn't see QPPs anymore. We just got noise out. So that was really cool. And, I don't want to say that I think the LC is necessarily driving these because all of these neuromodulatory systems are interconnected. And so I think we've basically taken a hammer to the neuromodulatory systems and we broke it. So I think it's involved, but I think we have a lot more work to do to kind of figure out, where that's going. Okay, so I haven't talked at all about machine learning yet because this was a long-winded way to give you the framework. But we found something exciting in our data, and it gives us new information about the brain that we didn't know before, that we couldn't see with anything else. And so we wanted to figure out what else we could learn from this data, and that's where we turned to the machine learning. So we started out with the simplest approach we could think of, which takes these resting state fMRI scans and looks at each data point as a single time point in a highly dimensional space, where the dimensionality is, defined basically by the signal in each ROI at that time. And then we did this reduction algorithm, this t-SNE, that takes that high dimensional space and uses a gradient descent to kind of minimize the divergence between that space and a two-dimensional representation of it. I've got a fruit fly on here because Gordon Berman gave us this idea with his awesome work on fly behavior. So we did that, and the cool thing is you can make maps. This is again with the Human Connectome Project data. I haven't mentioned it, but they also have some task data involved, included with that data set besides the resting state data. And we threw all of that together when we did this dimensionality reduction, and we were able to show that resting state data had different state distributions than task. So it's not like we were just pulling out unconstrained tasks. There's actually something fundamentally different going on. But more excitingly, when we started to look at the details of this, and I'm showing an example from two variations of a single task down here at the bottom, variation 1, variation 2, you can see that the points seem to move a little bit at different times after task onset. And so we started to think, well, maybe there are more trajectories in here that distinguish these very similar processes that we can pull out. So there's a technique that we thought might be useful to look at this called a variational autoencoder. And you guys might be familiar with this. You can think of it as a denoising network. It takes your input, crunches it down into a bottleneck where it has a very low dimensional representation, and then reconstructs a denoised version of your signal. But what we were really interested in is what happens in this bottleneck. Could we take our resting state fMRI data and crunch it down into a low dimensional representation in terms of these spatiotemporal trajectories? So this is an early example, and it's color-coded here by, this is again, weight in dimension 1 versus 2, and we're color-coding versus which network is dominant. But what I really want you to see is over here where we've plotted the course of a scan. So we've broken it out of all these dots and connected them together so you can kind of see how it evolves. And there are hints of trajectories. There are common loops that kind of go parallel to each other. Like maybe the same sort of thing is happening over and over again. Now, this was from one of our original implementations. It's not the one we went with finally, but I think that's kind of a good demonstration of what a trajectory might look like. Here now are some of the actual latent variables that we've been pulling out using the full network that I showed you with the autoencoder label. And so each of these now is one of the latent variables. We've represented all of this data as a combination of these 32 spatiotemporal trajectories. Each area is along the vertical axis, and its evolution over time is along the horizontal axis. And if we look at the primary trajectory that explains most of the variance right here, it looks just like that QPP. So that's great. We're getting the same thing when we analyze the data different ways. That's always encouraging. But I think it's also exciting that there are other trajectories in here to explore. Now, some of the ones, especially, down here that explain less of the variance in the data, may or may not be important. Maybe we didn't need this many latent variables. This is something we're still working on. But definitely there's enough interesting trajectories to explore, even in these first two clusters, that explain a lot of this different variability. Okay, so that was one thing we did, looking at trajectories and doing this data-driven approach. But we also want to be able to understand the trajectories that we see in the brain. And so I'm going to go back to that brain network model that I talked about a little earlier when we were talking about this sort of structural resonance hypothesis for the QPPs. And as it turns out, there are many, many neural mass models you can use, many ways you can build these models, and many, many ways you can parameterize these models. And if you use your average functional connectivity to parameterize them, the problem is almost all of the models do a good job. So while this tells us that these functional connectivity patterns can arise from these types of models, it doesn't really help us distinguish between which models are more likely to make those types of dynamics. And so we did our QPP analysis and some other types of dynamic analysis, like I showed you before. And we found that these are more sensitive to the type of model and the parameterization of the model. So here's an example where we did the same QPP analysis on the same model, but one had a low coupling parameter that weakened the coupling between areas, and one had a higher coupling parameter. And you see that if we want to get this sort of two-state QPP, we've got to go to this higher coupling parameter. So that made us feel like, these dynamic metrics might give us a good way to distinguish between the models and the parameterizations. But we still have this huge space of parameters to be swept, and we thought maybe machine learning would be a way to do that. Now, I want to leave some time for discussion, so I think I'm going to skip over this particular approach. I'll just give you the highlights. One way we can do this is to use a long short-term memory network, so it's a type of recurrent neural net, to try to fit the initial conditions for one of these models and then see how well that predicts. And we're showing it here on an example. We found that if we did this, it was sensitive to the type of model we put in, which is a good thing, so it can help us distinguish between parameterizations. And if we apply this to resting state data, we found that if we looked at how the structural network changed as we put this in, we could get very good distinction between the different parameterizations of that model. Okay, but I'm skipping that part because I want to get to this part because I think this is the most exciting part. The other thing we've tried is, instead of just learning the initial conditions, is to learn the parameters of the model at each point and basically get an ongoing estimate of those parameters of the model using the same sort of recurrent network approach. So we've done that now for a couple of different models. This one at the top is a simple firing rate model, and what you should pay attention to here is this is our actual data, this is our estimated data, and they look very similar. And then we also can show the latent states. Here for firing rate, it's not that interesting because it looks very much like the bold signal. But here's a model I'm really excited about because it includes excitatory and inhibitory components. So you can see again, we have the real resting state data, the estimated data that the model puts out, but now our latent variables tell us about excitation and inhibition. So hold on to that because I'm going to come back to that in a minute. So when we do our dynamic analysis, we find that this does indeed give us better representation of the QPPs than what we got with the traditional approaches. So I'll leave that. Okay, so where we are now in terms of machine learning, I think we can see that this has a lot of potential for extracting more information, like defined brain trajectories that we don't know about yet. So it's good for data discovery for these enormous data sets. I think this might also be a good way to get rid of some of these parameter sweeps and improve the accuracy of our models. And I really hope that these approaches will help us find more new things like this quasi-periodic pattern that we found that we wouldn't have known about otherwise. But I think that you always have to be aware of the caveats as well, because I think black box applications don't always tell you anything. I mean, if we didn't force our LSTM to learn the coefficients of a model and just let it learn the model, I don't know how to get something interpretable out of that. And the other thing that I want to point out that I haven't emphasized at all with this talk is that you have to be careful with the data you use. If you put garbage in, you're going to get garbage out. And for us, for resting state fMRI, Garbage means data that's contaminated with things like motion or physiological noise that kind of buries the neural signal between this other stuff. So, I think where we need to go with this is really tie this to multimodal methods and behavioral performance. And, be careful about looking at reproducibility. And the other thing that, I think is worth mentioning is we don't have to do machine learning for everything, even though it's, a hot topic and it sounds cool. Sometimes you just need a simple tool, and something like PCA works just fine. Okay, but this is the last slide that I wanted to get to real quick. I mentioned that excitatory and inhibitory activity that we were estimating from this non-invasive measurement of the resting state fMRI signal, right? Question is, how do we know if it's any good? How do we know if that's realistic? I mean, if we could get this information out of the bold signal, it would be revolutionary. But how do we know? And so we're currently setting up a method in the lab to do simultaneous optical imaging and MRI. And the cool thing about optical imaging is that there are genetically targeted calcium indicators that we can use to look specifically at excitation or at inhibition, get that data along with the resting state fMRI, estimate what we think they should be using that resting state fMRI alone, and then see how close we are. So then we have a way to validate our model or to prove that it's terrible, which might happen, but at least we'll know. And so I'm really excited about where that's going. Okay, so we have some resources available. We share our code if anyone would like to use it. We share data, and I've also put the link for the HCP data here. I just want to acknowledge the lab, especially Banaz, Amrit, and Jaudi, who did most of the work in this particular talk. And this is an old picture, but our new pictures don't look as cool because we all have to sit around on the lawn and wear our masks. So there we are. But thank you all for listening. I hope I've managed to leave a little bit of time for questions and discussion.
Thank you so much, Shelley. Here's some applause from everybody. All right, so let's open it up with questions. If you have questions, feel free to put it in the chat or just unmute and ask. If there's too many people, we can do the raise hand. And maybe I'll start with a question while everybody else is getting ready. So I really like the finding with the rats and the locust. How do you pronounce that? The LC, the locust gorillas.
I just say LC.
LC, perfect. And let me try to see if I'm seeing if I'm getting the interpretation right. So I think many of us in human neuroscience sort of think that these correlations in the resting state are mostly driven by the white matter tracts, but if you inactivate LC and they go away, that sort of suggests that it's not the white matter tracts within cortex, it is just neuromodulation from subcortical areas. Is that a correct interpretation of your findings?
That's what we think. It's not that we're saying that it's not the white matter tracts, but there is other stuff. The white matter tracts alone, we think, are not enough to account for these dynamics that we see.
Okay, so you think it's a combination of both, but maybe the neuromodulation is the larger determinant of the correlations.
Yeah, and that actually fits with a lot of work that people have done looking at changes in this resting state functional connectivity as people become sleepy, right? Mm-hmm. So if people are falling asleep, those changes show up as some of the biggest changes in that time course.
Yeah, that makes sense. All right, so we have a bunch of questions already in the chat. You can probably read them yourself, but I'll read them out loud for everybody. So Alice is asking, thank you so much for your talk. What changes to keep the pace do you expect to see in disorders like attention components, like with attention components like ADHD and autism?
Great question. So ADHD is one of the few disorders we've managed to completely investigate this in now. And what we saw is that the QPPs contribute less in patients with ADHD, which is consistent with this idea of a decreased input from these neuromodulatory systems. So it fits, which is really cool.
Yeah, perfect. I think Annabelle is just having a comment, wrote a comment that is really cool. So Nigel has a question about the subjective components or subjective correlates of QPP. Like, has it been found in other psychometric tasks with fluctuations in performance that require persistent vigilance? If so, is there a clue about the cognitive behavior implications might be?
Okay. Yeah, sorry. It took me a second to find it and follow all of that. Um, yeah, so we're looking at this, and we do a lot of work with Eric Schumacher to try to understand this. And so that psychomotor vigilance task that we did with Eric um, showed that there's definitely a relationship between the QPP phase and reaction time. We have more recently been looking at other types of task data. We've looked at the Human Connectome project data because they have working memory, but we think that's suboptimal because they do the task in short blocks that kind of overlap with the QPPs. And so we've more recently been analyzing some data that Eric's group acquired where people do like an in-back task continuously for 10 minutes without blocks. And looking at the QPPs and that, we are seeing some changes. We're still trying to kind of summarize that and parse it out. But there are definitely some things going on when we do this continuous performance that we weren't necessarily seeing in the block data.
So maybe let me just follow up on this, and I don't know if Nigel meant that. Do you think if you're able to ask people right after a QPP happened, hey, what did it feel like? Like, what were you thinking? What were you doing? Would they actually be able to tell you, hey, this in the last 20 seconds, this is the internal thing that I went through?
I don't think so, just because this is going on all the time. You know, this is like a constant background activity in the brain. And so I think, and I don't know this yet, this is just kind of a working hypothesis, but I think that if we have times when we're very alert and focused, we're probably going to see very strong, frequent QPPs. You know, it will come out of the background activity and dominate the signal more. Whereas I think if we are relaxed and not focused, like when we're falling asleep in the scanner, we'll probably see weaker, slower QPPs. But that's kind of educated guesswork at this point, and we're still trying to figure out some of the analysis to do to see if that's true.
Also, I know your last question from Bilal. Have you thought about using some simple pharmacology to block or enhance QPPs by modulating glutamate or GABA or norepinephrine?
Yes, yes, a little bit. And we would love to have more ideas on this. I'll tell you what we're doing at the moment. As you know, I told you about the DSP4 study, which basically kills off those LC neurons. We're trying to do something that is a little more subtle, and we're working with David Weinschenker's group to go in and optogenetically stimulate the LC to see what the effect on QPPs is there. We have the pilot data from that, but we don't have the analysis yet. I can tell you we're seeing some changes in something, but I don't know yet if it's the QPP. But I know there are a lot of tools for manipulating these types of activity out there, and I would love to learn more.
All right, so it's 12:16. So if somebody, if whoever needs to go can go, I guess. But maybe you can just go through the remaining questions. So Ronnie just posed a question, and maybe there'll be some more. So, uh, Ronnie, do you want to unmute yourself actually and ask it? I feel like it's probably best if the questions come from the people who ask them rather than me trying to read them.
Sure.
I
Can you hear
don't
me?
want to put you on the spot. Okay. Um, so, so Ronnie is saying, I've been getting into ML techniques recently too for your dimensionality reduction techniques. May I recommend using UMAP in addition to the temporal SNE or TSNE, as UMAP seems to preserve more of the data's global structure?
Yes. Oh, thanks for the suggestion. At the time, we worked with TSNE because that's what we knew about, but I know people have come up with some better algorithms at this point, and that, definitely I think going forward we'll check that out. Are there any questions I missed? I know Adobe's doing a great job of keeping up with the chat. I keep getting lost in it myself.
Yeah, I think we've covered everything to this point. All right, so last call for questions. You can also unmute if it's going to take too long to type it. All right,
Well,
we'll
thank
see
you
you.
all for listening, and please, contact me if you have ideas or suggestions or things you want to talk about. I'm always happy to talk science.
Awesome. One last round of applause. Thank you, Sheila. Join us again next week, same time, same place, I guess. Bye, everybody.